Hi everyone, welcome to this webinar. I'm Fangyi Chu, the Technical Account Manager from Sino Biological. In this next 30 to 40 minutes, I will talk about the role of transmembrane proteins in cancer biology and introduce a groundbreaking solution by Sino Biological that revolutionized transmembrane protein production, catalyzing advancements in oncology research. For this talk, I will separate into 4 parts. First, I will brief overview what transmembrane protein is and why it is important. Later, I will introduce our new platforms for generating transmembrane proteins. After that, I will show some case studies that Sino has produced and listed on our website. In the end, I would like to talk about making bispecific antibody to target transmembrane protein to immune response. So for the first part, cells and organelles such as mitochondria or endoplasmic reticulum has membrane to separate cell interior from the outside environment. The membrane consists of a lipid bilayer with lots of membrane proteins, which either attach to or associated with the membrane. The membrane proteins control the movement of a substance in and out of a cell. Additionally, membrane proteins are involved in a variety of cellular processes, such as signal transduction, cell adhesion, and cell-to-cell interaction. 70 to 80% of the membrane proteins are integral membrane proteins. They are permanently attached to the biological membrane. There are 4 types. The alpha helical anchor protein only bound to one side of the membrane. The other 3 are transmembrane proteins. They are philopathic, which has hydrophobic regions, for example, alpha helix or beta barrel, that are embedded in the membrane. And there are regions that are hydrophilic, so can be extended into cytosol or extracellular environment. Other membrane proteins are either lipid anchored to the membrane or peripheral. Transmembrane proteins are important drug targets since they influence diverse cellular processes, from signal transduction to immune response modulation. Researchers have unlocked potential targets and designed small molecule drugs. Around 50% of the small molecule drug targets membrane protein. G-protein coupled receptors, GPCR, is the most diverse and largest group of membrane receptor protein in eukaryotes. They transduce extracellular signals into key physiological effects. Their endogenous ligands, including odors, hormones, neurotransmitters, chemokines, and varying from photons, amines, carbohydrates, lipids, peptides to proteins. Therefore, GPCRs have been implicated in a large number of diseases, such as diabetes, obesity, depression, cancer, and many others, making them attractive to drug development. And latest statistical data indicate that 527 drugs are approved by FDA, and around 60 of them currently in clinical trials. Usually, after researchers discover the target, they will perform structural study for drug design. However, it's hard to make the proteins and also lack of assays to validate the drug. The challenges in producing the transmembrane protein are that they are hydrophobic, so not soluble without detergent or lipids. Expression is also tissue-specific, and the exogenous expression might cause cell damage, leading to low expression. Even though we can extract the membrane protein from the tissue, the efficiency might be low. while the expression, the purification process in the exogenous expression might be complicated. The resulted yield is mostly low with low purity. In general, the transmembrane protein itself is hard to maintain the conformation and prone to aggregate. Thankfully, Sino has established 3 platforms to generate transmembrane protein, and I'm going to introduce those 3 platforms in the next chapter. So the first platform we have is the virus-like particle, VLP. And the second one is Detergent micelle platform. And the last one is Nanodisc platform. In the virus-like particle, VLP, it is a nanoscale particle, approximately 100 to 300 nanometer in diameter. They are comprised of viral capsid proteins surrounded by a cell membrane and membrane components derived from the host cells. This way, that the transmembrane protein can be displayed in their native conformation, so it's well suited for immunization and antibody screening. Also, due to lack of the viral genetic materials, the particle cannot replicate and it is safe to produce. Sino Biological has developed a VLP protein platform using the HEC293 cells. The second platform is micelle detergent platform. In general, transmembrane proteins can be solubilized by using detergent to disrupt the hydrophobic association and destroy the lipid bilayer. When we mix the detergent with the membranes, hydrophobic end binds to the hydrophobic regions of the membrane, thereby displacing the lipid molecule. As you can see here on the left, at the low detergent concentration. When the concentration is above the critical micelle concentration, then the monomers self-assemble into aggregates and driven by the hydrophobic effect. And the polar end of the detergent brings the membrane protein into solution as detergent protein complex. Micelles are typically a few nanometers in diameter and have a molecular weight of less than 100 kilodalton. How the micelle form in the solution affect the solubilization of the transmembrane protein. Micelles are widely thought to be spherical in shape. However, recently people have found that it's actually asymmetrical and have a rough surface. The tail are disorganized and poke into the solution. And the detergent micelles are also dynamic. The detergent monomers within the micelle are constantly exchanging with the free detergent monomers in the solution. Therefore, its stability is highly depends on the detergent and the concentration of the monomers. And the third platform is the Nanodisc platforms. First, one way to make the nanodisc is using SMA. SMA stands for styrene, maleic anhydride. Membrane solubilization typically requires the use of detergent, but this is not an ideal situation as the native lipid environment is stripped away by the detergents, which may lead to protein misfolding or aggregation. So a promising alternative is the use of SMA polymers. SMA inserts into cell membrane, and then we're cookie cutting the protein from the membrane. Proteins in the nanodisc can be purified and then characterized in their native lipid environment. In addition, it is possible to study the protein-lipid, protein-protein, and protein-ligand interaction using this platform. The advantage of this one is that there is no need to use detergent in the protocol, and the size of a nanodisc is around 10 nanometer in diameter. Another way to make nanodisc is by a membrane scaffold protein, MSP. The membrane protein target is first transiently solubilized by a detergent, and then adding the MSP and the phospholipids. When the detergent is removed by dialysis or absorption to hydrophobic beads, the target membrane protein simultaneously assemble with the phospholipid into a discoidal bilayer with a size controlled by the length of the MSP. The result, nanodisc, keep the membrane protein in solution, provide a native-like phospholipid bilayer environment that can provide stability and functional requirements of the target. The size is usually around also 10 nanometer in diameter. However, you will need detergent in this protocol. So now I'm going to talk about the case studies that Sino has produced in our catalog. The workflow of our transmembrane protein starts from gene synthesis and cloning. We then transiently transfect the plasmid to HEC293 system for expression. After 5 to 7 days, we will harvest and break down the cells to extract the membrane and solubilize the transmembrane protein based on different platforms. For micelle and nanodisc, we will further purify target proteins according to different tests. In the end, we will perform QC tests. Here, I list the catalog products we have produced using the 3 platforms. On the left, you can see the VLP products. On the right, you can see the detergent and nanodisc products. If you don't find what you need in these lists, we also can take customized projects to produce your protein of interest. Later, I'm going to show some case study for these catalogs. The first one is GPRC5D, G protein coupled receptor family C, group 5, member D. It is highly expressed on the surface of the CD138 plus multiple myeloma cells, while it's low expression in the normal cells, rendering it a promising marker for tumor load and for immunotherapy. We validated the GPRC5D VLP product by ELISA against the anti-GPRC5D antibody, and the batch-to-batch consistency is high. And also, we validated the VLP protein by Western blot against the flag tag. On the other hand, injecting the VLP in the animal for generating antibody also gives higher titer than the conventional DNA immunization. So this is a very good antigen platform also for generating transmembrane protein antibody. Then for the detergent version, it was validated not only in ELISA, and also bilayer interferometry, BLI, to measure the affinity constant. The KD is around 58 nanometer against the anti-GPRC5D antibody. And Sino mostly use SMA to generate nanodiscs. The GPRC5D SMA nanodiscs show binding against the antibody in ELISA, BLI, and also the surface plasmon resonance, SPR. The affinity KD can be as low as 0.7 nanometer, which is lower than the result from the detergent platform. So both of the detergent and nanodisc show very good activity. Another case study I would like to show is the clouding proteins. Clouding proteins are tetrastrand transmembrane proteins of tight junction between the cells, which modulate the paracellular permeability, polarity, and signaling. Claudin 1 and Claudin 6 proteins are involved in HCV entry, while Claudin 18.2 is highly expressed in tumor cancer. And Sino has both Claudin 6 and Claudin 18.2 in stock, and we are currently generating more Claudin proteins in the family. Here, both Claudin 6 and Claudin 18.2 VLP and detergents are validated by ELISA binding to the corresponding antibody. And the nanodiscs show KD to be as low as 0.5 nanometer for Claudin 6, and for Claudin 18.2, the KD can be as low as 4.3 nanometer, showing that both our nanodisc proteins are active. After showing our platforms and some case studies of the transmembrane proteins we produced, I would like to reroute direction to the anti-transmembrane bispecific antibodies. As I mentioned before, transmembrane protein is a very good target for immunotherapy. Researchers could design bispecific antibody to redirect the immune system to attack the transmembrane-related tumors specifically. There are published sequences of the anti-transmembrane protein antibody available for design and for recombinant expression. In the meantime, Sino is also currently developing anti-GPRC5D and anti-Claudin antibody using the VLP and nanodisc proteins we have in catalog as an antigen. In the last part, I would like to introduce Sino's capability on producing bispecific antibodies. Sino has experience on more than 14 bispecific antibody formats in the HEC293 system. You could see the chart here and the illustration here. So based on this chart, Dutamab, IgG, SCFV, Nobinhol, and BITE are common format. Until now, we have completed more than 80 projects with averaging over 90% success rate, and the bispecific antibody can be validated in cell-based assays and by SPR-BLI. Our highest yield is 250 milligram per liter with larger than 90% antibody purity. Here, I'm going to show some case studies. First is the IgG SCFV format. The SCFV is on the heavy chain of an IgG. Usually, we can get high purity after protein A purification. By SDS page gel, you can see it's larger than 95% purity. But when we run the SAC HPLC, the purity might not be high for the monomer. In this case, it's only 72%. So we perform gel filtration to isolate the monomer peak to reach 98% monomers. Usually after 2 or 3 steps of purification, we can always get very pure monomers. The last example is SCFV with Nobinhol design. From the SS page gel, we can see that the molecular weight of the Nobinhol, SCF knob and SCFV hole are very similar. And there are aggregates and potential mismatch showing in the SAC HPLC data after protein A purification. So the mismatch means that the knob or knobbing hole or hole to hole, while our target antibody is knob and hole pairs. But luckily, the isoelectric point PL of the pairs are different. PL of hole to hole is larger than PL of knob to hole and larger than the PL of knob to knob. So we can separate the target proportion by ion exchange. So here you can see the ion exchange profile. So we can further purify this proportion knob hole from the sample. And then after that, we can run the stack HPLC again to get rid of the use the gel filtration to get rid of the aggregates to get over 98% of the monomers. So here, just showing how Sino usually produce and purify the bispecific antibody. And this is our protocol. And we can definitely be flexible to change based on clients' needs. And usually we can get very high purity of the bispecific antibody. So to summarize my webinar today, we have discussed 3 platforms for transmembrane protein production that Sino has established. Based on the ELISA, SPR, and BOI data, the binding activities are good for all platforms. However, currently, VLP is not suitable in SPR and BOI tests yet. We are optimizing the testing condition. And the VLP platform takes shorter time in production, but the final product is actually a mixture of the target transmembrane protein and other viral proteins. Detergent and Nanodiscs platforms can get pure target proteins, but take longer time and cost more. Another thing for Detergent platform is that we cannot directly coat the detergent micelle on regular ELISA plates, but we have to use the nickel-coated plates. For the recombinant antibody production, I mentioned the bispecific antibody. Sino, of course, can produce regular antibodies from different species, including human, mouse, rat, and rabbits, even Sino, and also with different isotypes and subtypes. Other than IgG, we especially expertise in IgM and IgA production. Other than those, the fragments such as scFv, Fab, Fab2, nanobody are also available. What we need is just a sequence, and then we will synthesize a gene and clone into the vectors for transfection. And for the full-length IgG or IgM, IgA, we co-transfect the heavy chain light chain with J chain to get the oligomers. In our recombinant technology, our advantages are that we are able to produce more than thousands of antibody at once. Also, we have up to 1,500 liters of bioreactors, rendering us to produce kilogram level production. Also, all the reagents are 100% in-house with 5 different expression systems. So we have a large, diverse, and variety to produce any protein that of your interest. Our recombinant production includes proteins, high-throughput antibody, large-scale antibody, chimeric antibody, also fragments, Fab, scFv nanobody, and also bispecific and humanized antibody. So basically, just let us know the sequences you have, and we can evaluate which platform to use, what's the purification method to suggest, and also different tags. Sino Biological is a one-stop reagent and CRO service provider. We have more than 6,500 proteins, 14,000 antibodies, more than 47,000 genes, and more than 600 of ELISA kits. Our CRO services cover recombinant expression for protein, antibody development, and customized assay development. Here, our certificates are shown, and our products have been featured in many high-impact journals and spread footprints in more than 90 countries. We truly hope that our effort can help you work and research to exert more and more beneficial influences on the scientific and human health field. We have offices all over the world. In the US, we have a warehouse in Pennsylvania for all the US stocks, and we also have an office in Texas. We are also going to open up a new production site, Center of Bioprocessing, in Houston next month. Therefore, in the future, Sino will be able to provide services locally with faster turnaround time and easier communication. If you're interested in learning more about our new facility, please let me know. Thank you for joining me in this webinar to explore the world of transmembrane proteins and learn about their role in cancer biology, and also discover how Sino Biological's cutting-edge services can catalyze your research journey. Hope can hear from you soon.